Chytridiomycosis, or chytrid for short, is a disease that affects the skin of amphibians. It's a fungal infection caused by Batrachochytrium dendrobatidis or Batrachochytrium salamangivorans, known as BD and B-sal respectively. It's a waterborne fungus that latches onto the skin of an amphibian, but only causes minimal, usually unnoticeable symptoms. The cause of death is usually heart failure, but an infection does not necessarily guarantee death. Chytrid started spreading in the 1970s. It reached pandemic levels. It spread really fast and killed huge amounts of amphibians. It caused major declines in amphibian populations and huge biodiversity loss. That kind of thing can have widespread consequences on an ecosystem, and disrupted food chains and unfulfilled niches can have impacts that are difficult to predict. It's important to monitor local amphibian populations to make sure chytrid isn't spreading at the rate it was at its height. The Advances in Herpetology class went to Newborough Forest in North Wales to perform our own chytrid survey in March 2025. We collected palmate newts, great crested newts, common frogs, and common toads. Amphibians are mostly nocturnal. Our survey had to be conducted at night so that we had better chances of actually finding and catching amphibians. We tested each captured amphibian by swabbing its belly and feet. After swabbing, each frog, newt, and toad was safely returned to the spot where they were captured from. The swabs were kept on ice until we were ready to test them for chytrid. Back in the lab, the DNA from each swab needed to be separated from the rest of the sample. After that, a qPCR was performed on each of the samples we brought back from Newborough Forest. A qPCR, or quantitative polymerase chain reaction, separates a specific section of DNA from the rest and copies it hundreds of times. In this case, the presence of that specific section indicates a positive test result. No chytrid was detected in any of the 56 amphibians swabbed. This is great news, but how does it compare to past chytrid surveys? The results of a 2008 chytrid survey showed that 66 individuals tested positive for the infection across 19 sites out of 121 total sites. However, there are some differences between this survey and our survey that are important to note. For example, the 2008 survey had a much wider study range, taking samples from multiple ponds throughout the UK, while our survey took place in one location. They also paid multiple visits to the same site. They surveyed in April and May, then again in June and July. So why does this matter? Even though chytrid was detected across the UK, there were many sites that did not have any infected individuals. While there were no amphibians with chytrid at the Newborough Forest site, that does not rule out the possibility that there are infected individuals elsewhere. Also, our survey was conducted slightly earlier than recommended by standard protocols. Taking samples at the same time as other surveys eliminates a variable and ensures that the results are comparable across surveys. That 2008 survey was part of a citizen science program. Volunteers across the country participated and swabbed over 6,000 amphibians. Not only did this project collect valuable data, it also helped to raise awareness and get people interested in amphibians. I hope learning a little bit about the field and lab work that goes into studying amphibians inspired you to keep discovering new things about the wide world of environmental science. Thank you for watching.